So in a cell's lifetime, it goes through many, many, many cycles, okay? And the dividing part is only one little step of the cycle, okay? So it is not dividing over and over all the time. The division is only one part, okay? So the rest of the time, the cell is in something called interphase. So we're going to go into more detail about that. But on your note outline, it says during the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells. Now, I cannot tell you why they are called daughter cells, but they are.
Because they're all girls.
They're not all girls.
Isn't it the male that gives gender to in creatures?
Yes, but that is true, but I don't understand why that would mean that they're called daughter cells.
Girls are the
Doesn't
ones
make a
that
whole lot of sense.
guys can't.
Apparently girls are the most important thing in both types.
They're the ones that get the birth.
Okay, whatever.
Okay,
Guys don't.
let's focus, shall we?
Got me.
So in the cell life, it goes through this cell cycle. Now we're going to jump down to where it says the eukaryotic cell cycle. We will go back to where it says prokaryotic cell cycle. For most of the time, we are going to be talking about what eukaryotic cells do. The cells with the nucleus, plants, animals, all those kinds of cells. So in this cell cycle, the longest phase is called interphase. That is basically when everything else other than division is happening. And on these pictures, it shows as this very long 3/4 of the time phase. Okay? But then it is broken up into smaller phases. Okay? So what you have listed are each of these, correct? G1, S, G2. Okay. So let's start with G1. I like to think that G stands for growth. I don't actually like that it's used to call gap. I think that's weird. That doesn't make a whole lot of sense to me. Growth, I can wrap my head around. So this is from the beginning, and again, the cycle goes over and over all the time. But the first part of interphase is called G1, and that's where the cell is growing. That's easy to remember. Okay, then in the S phase, the S stands for synthesis. Now this is easy to remember because synthesis, we just talked about protein synthesis means making something. Okay, well if you keep remembering that synthesis means making something, then it's easy to remember that during the S phase, the DNA makes a copy of itself. Why would the DNA need to make a copy of itself at some point?
So
It's gonna divide.
Okay, because why? When it divides, what's going to happen?
It needs to have DNA in that cell.
Absolutely. So there needs to be DNA in both of the cells. And right now, the cell will only have one copy of the DNA. And so before that division occurs, the cell needs to make a copy of the DNA. And that happens during the S phase, or synthesis. Okay, we're still in interphase. Now we're going to the second growth phase, or G2. The cell is still growing. Remember, it doesn't divide until it's nice and big and fat. Yes, sir?
So this is going up.
This is going up. What do you mean?
This question and this question.
I have to see.
Oh yeah, we're going.
Oh, okay.
You're under eukaryotic cell cycle. We're going to go to prokaryotic here in a minute, but really we're going to spend most of our time talking about eukaryotic cells. So the second growth phase, and then in that point the cell is preparing for mitosis. That feels a little vague to say, but we're going to leave it vague for now until we understand all of mitosis. And then the exciting part is the M phase. The M stands for mitosis.
cell splits.
Yes, specifically mitosis means the nucleus is dividing. Okay, but then right after the nucleus divides, the rest of the cell splits too. And then we will go into all of the phases of mitosis. But mitosis is technically the division of the nucleus. When the nucleus divides, just like we said before, each new cell is going to need a set of DNA so that they are able to have all the information about being that kind of cell. And the cells that are created are identical. Okay, so you have all your cell cycle filled in, G1, S, G2, and M. Okay, let's jump up from from there and talk about how prokaryotic cells do this process. It's kind of boring, I hate to tell you. A prokaryotic cell doesn't have a nucleus. So they're not going to go through mitosis because mitosis is the division of the nucleus. Okay? We already talked about that their DNA is just kind of free floating in the cytoplasm. And instead of lots of chromosomes, which we're going to see later when we get into mitosis, most prokaryotic cells just have one chromosome. It is one circular chromosome. It's usually pretty long and kind of twisted up, but it's really just one chromosome. Okay, so where it says prokaryotic cell cycle, I would probably put something, no nucleus, single circular chromosome. And then we'll see how it divides. It's real easy. No nucleus, single circular chromosome.
What do we put for binary?
We'll get to that. Just it's almost there. Next slide. Yeah, we're right here. We just did all that.
It's not a phase, it's real mean.
Now we're right here. No nucleus, single circular chromosome.
The chromosome's like a rubber band.
You think?
Yeah.
Okay. So then the prokaryotic cell cycle, we see similar patterns where the cell grows. The DNA has to copy. That's a fancy way of saying copy is replication. And then the cell divides. So similar process or pattern, but there's not all of these phases like we see in a eukaryotic cell. But I would like you to write underneath where you just wrote no nucleus and single chromosome. Growth, DNA copies, and then the cell divides. The cell division in prokaryotic cells is called binary fission. It is not called mitosis. It is called binary fission. I'm pretty sure there's a picture on the next slide. Nope, there's not. So in binary fission, you'll usually see a picture like this, and then a picture of the cell with two circular chromosomes, and then the next picture has two cells. That's it. So it's real quick. I can draw it up on the board, but it is just once this chromosome copies itself, the cell divides, and it's called binary fission. So usually, once the DNA is copied, it looks like two copies. And then it'll usually just have an arrow, and then one cell with one circle, and then another cell with another circle. And that's it. It's very short and sweet. So for binary fission, I would just write prokaryotic cell division. For binary fission, just put prokaryotic cell division.